Okay, good morning, everybody. We are today for the round table for the workshop that will be very interesting. And so we will start with the round table, and I would like you to just to remember our speakers of yesterday so that you know that you can make questions and observation. Remember our eminent scientists that talked yesterday. So Professor Laura Lieto, University of Naples, Joe Polik that will join us in 10 minutes, so later. University of North Carolina, Wilmington, and then we have William Gervich, University of California in San Diego, and Vitor Vasconcelos, that is at University of Porto, also the president of the Center for Marine Research in Porto. So, and finally, Mauricio Caraballo Rodriguez, University of California in San Diego, that also will share his knowledge about what is possible to do with this new advanced technology to use mass spectrometry as a tool, a very powerful tool detecting molecules. And so I will open the discussion now, and you are invited to introduce yourself and make your questions. Okay? Um, so who would like to start? Maybe we have a list, Germana? Mohamed, would like to start? Good morning. Non li vedo. Ah.
Can you hear me?
Yeah, oh, yes, we can hear you. Good morning, Mohamed.
Good morning
Nice
to
to
all
meet
who are in
you.
New York and evening who are in Europe. I'm first year PhD student and I'm speaking from Tübingen. And I'm working on research on natural product chemistry with a special focus on Nocardia species at the moment. My question is a bit different from that marine things. The question is that could you please suggest me some effective extrinsic methods or co-culture strategies to activate biosynthetic gene cluster, because in Nocardia, I can see their genomes, they have great potential, but in laboratory, they are unable to produce something.
Did you get the question? No.
It was a little hard to hear.
Yeah. Can you repeat your question? Maybe just a bit slower because we have
Okay.
some problem in hearing you.
My question is that I'm doing research on Nocardia species, and their genome has a great potential. They have it's a full, but in the laboratory when I try to get secondary metabolites, they are unable to produce something. So silent biosynthetic gene clusters.
Oh.
So I am a chemist. I don't want to use intrinsic approaches but I want to try extrinsic approaches to activate the biosynthetic gene clusters. So could you suggest me some effective extrinsic methods or co-culture strategies, or especially in laboratory settings, to enhance secondary metabolite production?
Okay, so Professor Gervich is going to answer you.
Okay.
Good morning, and great question. Two thoughts come to mind, although I'm not completely sure what you mean by intrinsic and extrinsic methods, and maybe we can discuss that. But the two thoughts that come to mind are, one, there is this fairly well-known approach called OSMAC, one strain, many express compounds. And so varying the conditions of growth of your Nocardia species may upregulate the expression of certain pathways. But this is a long-standing question in the field of natural products chemistry. And a person that you might want to look at the work of is Mohammad Samadost at Princeton University, who has taken a rather methodical approach to finding small molecules that will turn on pathways in microorganisms. So he has published some wonderful papers there, and there's some, I think, some very good clues as to what you might try with your Nocardia species. So with those two answers, maybe clarify what you meant about intrinsic and extrinsic.
Can I speak, or it's somebody else's turn to ask the question?
Somebody else would like to add some comment on this? Maybe
I
le-
can ask,
yeah.
but I think that will. Mohammed, before I say some suggestions, Bill asked you about elaborate a little bit more. What do you mean by extrinsic versus intrinsic approach?
I mean,
Because that is not completely clear in this context.
okay, intrinsic, I mean, some people they do heterologous expressions or genetic changes by making these things, and extrinsic, I mean, maybe co-culture with some things, maybe other bacteria, or recently I'm planning to do co-culture with the human cell lines to give the initial conditions. So maybe because Nocardia are biosafety level 2, and so this is in my mind, and in that sense, I was talking about intrinsic rather these biotechnology related approaches, or extrinsic like co-culture, or maybe with the human cells or other microorganisms.
Can I say something more, Bill? Or?
No.
Okay. I can say something. Mohammed, based on your question, then let's say you're working with pseudonocardia, and then you say that it's interesting because it has a lot of potential as a prolific producer of molecules of interest. Well, the first thing that I would ask is where these pseudonocardia strains that you are working with are coming from. What was the natural environment when you found or these strains were isolated? Let's say they are associated to insects, or they are associated to plants, or they are from marine environments or something like that, then you might start thinking what would be the nutrients or resources that this strain would have in the natural environment to outcompete other microbes. Because with that, you might suggest which culture media or what kind of compounds you might to mix into the culture media to induce the production of some silent biosynthetic gene clusters. The other approach that I would suggest is if you have the genome sequence, which are the biosynthetic gene clusters that you are going to focus on? Are they really unique, or they have been found in many other strains? Because you might spend the next 5 years of your research working with some molecules, but yes, the novelty might be interesting, but why would you focus on that one or two molecules? And then you can see which ones you would select for future experiments. From the ecological point of view, I think that that's interesting. And of course, applying some mass spectrometry, as I told you, like the replication approaches, then you can say, okay, I detected these molecules in these conditions, but which ones are you going to focus for further structural elucidations? Are they unique or not? There are several approaches you might think of, but yeah, I can give you those comments.
Okay, can
Thank you.
I ask?
Thank you so much.
Okay.
Yes, please go ahead.
Okay, I have another question, if you allow me to, regarding molecular networking, because when we do molecular networking, I mean, by using this software, we can abstract the masses which are produced by the media and which are produced by our strains. So we can get idea that, okay, this mass is only produced by our microorganism, and this mass is not produced by the bacteria. And obviously after that we have to decide, we have to work on HPLC for to isolate that compound. So how much we can rely on these softwares like GNPS and other tools, that how much we sure that we can rely on them, and then we can continue our study and we can expand our project or cultivate on the larger scale and start isolating.
Hey, Muhammad. For your question is, how much can we rely on these softwares, right?
Yeah,
So
exactly.
it depends. Just for starting, when you use mass spectrometry, you assume that you extracted something from your experiments, let's say from your cultures. You do some extractions. I'm pretty sure that you're missing something, because what you extract is soluble in your extraction solvent, or depending on the approach. Then you go to the mass spectrometry, then you ionize the molecules, but not everything is ionizable. Depending on the efficiency of the molecules to ionize, depending on the instrument, etc., you might be also missing something. And then you go back to the genomes and the genomics analysis, then you say, okay, which of the biosynthetic cluster were actually expressed, and which ones molecules were actually produced and detected. You might have been missing something. So even that is not perfect, with the information that you are able to get from tandem mass spec, there are several tools, but just starting with molecular networking, you might see which masses, for instance, are unique to the specific condition that you have. And that is a starting point. Every single fragmentation spectra that you got that correspond to a potential molecule is present in any libraries, or is present in another data set of microbial molecules. If so, then that gives you also some insights. Are the molecules that you detect in your experiment also present in other pseudonocardia's data sets? Then you can rely on this. There are a lot of siderophores that are pretty common in actinobacteria and actinomycetes. Then if you detect those, then you are confident that the approach that you are learning, because it's probably something that you will learn during the process, is working for you. And then you have some reference molecules that might be used also to guarantee that the conditions that you're using, the method that you're using in the LC-MS are working. Then you have those reference molecules to be sure that what you are detecting is reliable, and so on.
Thank you very much.
Okay, thank you for joining us. Hopefully, you get some more clues on what your research. But you can also send a message on our email, info email, and we will try to give you some answer. Okay, thank you so much.
Thank you very much.
Bye.
You know, it would be fun to know where the students are located as they introduce
Tübingen
themselves.
is in Tübingen,
In Tübingen.
Germany.
Okay.
Okay, so the next participant. Anna, are you with us?
Can you hear me? Okay,
Yes.
great.
Hi, Anna.
Good morning,
morning.
everyone, because there is a morning. I'm a PhD student of the University of Catania in Italy. And in Italy right now is evening. So I'm an organic chemist working on natural products, in particular from agriculture. I'm trying to recover bioactive compounds from different kind of agricultural and food waste. First of all, I would like to thank all of you for the brilliant talks and the really inspiring knowledge that you share with us. And now the question is concerning the talk of Professor Jerwick in particular. That was I was inspired by the fact that the majority of pharmaceuticals right now are deriving from natural sources, right? Like the marine environment and plants, as all of us know. But my question is, do you think that a kind of biodiversity that is liking in the future we expect these decrease in biodiversity. Do you think that a new source, kind of new source of biodiversity, can derive also from byproducts and waste? Thank you.
Bill, this is a question for you.
Yeah. I'm just thinking about that. New biodiversity from the byproducts and waste streams from agriculture. Was that was the question? Is that do I think that could be a valuable source of biodiversity for drug discovery?
Yes.
Yes. Okay.
If we can think about a substitution of the ordinary source of natural products.
Yeah. Well, I'm sure there's many answers to that question, but let me answer it the way that occurs to me right now. I would say, and although I showed a graphic for that for marine life, that it seems that most of the most highly bioactive compounds that we've discovered to date are actually deriving from the metabolic processes, from the genetic capacity within microorganisms. And so as you talk about agricultural waste, the thought that comes to mind is that that's food for microorganisms. And of course, the type of microorganism that you grow is going to be a function of the sugars and the fatty acids and the protein components of that foodstuff. So my mind naturally goes to using those in some kind of a fermentation use to select for particular kinds of microorganisms that then would express certain kinds of biologically active molecules. The agricultural wastes themselves, the food stuff, actually that's something that Mauricio, you might be able to answer, is that Peter Dorstein has had a ongoing research project, as I am aware, studying the chemistry of food in a much more in-depth way than has previously been done. So perhaps you could speak to that point.
Yeah. Thanks, Bill, and thanks, Anna. As Bill said, there are probably several ways of answer this, and I will give you two. Ones that Bill suggested based on the food projects that we have in the lab, but another one that occurs to me, and is a project that I worked with a collaboration a few years ago with the University of Connecticut studying fungus growing ants. Those insects that are able to culture a fungus as their food and their habitat. So they collect plants from the surroundings and then bring them to their fungus to grow. Then the fungus metabolize, degrades these plants, and then produce the nutrients that are accessible to the ants. So when I saw that, to me it was something that can be waste for some organisms is actually the food for others. So in that sense, is basically everything that we use as a food and then we just produce some waste should be ecologically recycled, used by others. It can be other organisms, bacteria, etc., to metabolize and then probably be a source of new molecules for several reasons. One of several projects that we have in the lab as well that is related to food is, and that's how I presented one of the links, is that how we curated those data sets and then we were able to develop food mast. So if you got a molecule, you will find it into food. But let's think about the diversity in terms of chemical space that is found in food. So imagine, and it's something that we're working on as well, imagine a burger. So we eat a burger. And there are a lot of microorganisms that are in our guts that help to metabolize and degrade all the molecules that are coming from the onions, from the beef, from the tomato, from the lettuce. All this chemical complex is also used by the microbes to transform in new chemicals. But if you start associating what is the benefit of every single molecule from every food, that you can, instead of looking for medicines, then you can start interpreting the chemistry of the food. Is this food used for you in this condition? For instance, in Alzheimer's disease, there have been some association of, let's say, blueberries as a healthy marker versus other foods that are not that good to decrease the development of Alzheimer's disease. If you can find those biomarkers in food that are associated
with
to a beneficial condition, then you can start teasing apart which molecules are coming from the food that are actually helping to your health. So instead of looking for drugs in the long term, we will probably go back to our ancestors that were using medicinal plants with the whole complex of chemistry in a plant, in a concoction that was used for a specific treatment, instead of looking for a single molecule to cure that disease. So that's how I see, and I think that is very complex answer, but it was a very complex question. So yeah, there are many other answers that we can think of.
Thank you.
Anna. Definitely, I think that the waste production is a new reusable source of new bioactives. In our group, we are working on the waste from the beer production, and this seems a very promising project as well. So there is a lot to discover, and what you are suggesting today is very interesting. So go ahead with this. Okay.
Thank you, everyone.
Okay. Thank you. Bye bye. Anna was also in our last summer school in Maratea, and so is following the community as well. So. Good morning, Sabrina. Or good afternoon for you, of course. Are you
Hear
there?
me? Can you see me? Yeah. Can you hear me? Can you see me?
Hi.
Can you hear
Go
me?
ahead with.
Hi.
Okay.
Hi. Yeah, this is afternoon. I'm Sabrina Giordano. I speak from Italy. I'm in Naples right now, and I'm a PhD student in biotechnology. So my main focus is peptide nucleic acid as an application for for developing new biotechnology tools. And my question is for Professor Jerwick, and my curiosity is: do you think it's possible to maybe change the metabolism of bacteria in order to, let's say, use them as a modulable machine in order to produce more oxygen?
So your question was to manipulate microorganisms to produce more oxygen. Do I have that correct?
In order to improve, in order to improve, of course, their use.
Well, interesting. I mean, of course, photosynthetic microorganisms like what ourselves and Professor Vitor Vasilius studied, the blue-green algae or cyanobacteria, they're producing molecular oxygen. and they were responsible for the generation of oxygen on our planet for billions of years. That initial oxygen was all absorbed into rocks to make various carbonate and other kinds of oxidized rock molecules. When that became saturated with oxygen, then we started to develop our current oxygenic atmosphere, 20% oxygen. So to increase that, I'm not sure I would think to use biotechnology to do so, but promoting habitats that harbor photosynthetic organisms that make oxygen would be positive. Of course, and much of that is occurring in the oceans, as I think you mentioned, that 50%, I think you mentioned, was coming from open ocean micro cyanobacteria. Much, of course, also comes from land plants and their photosynthetic processes as well. Open oceans have been looked at as kind of deserts that we might be able to manipulate and do things to change the productivity of those areas. This has been very controversial. The main goal of some of that has been, can we increase carbon capture in the oceans by providing limiting nutrients to those open ocean environments, like iron in particular. I've read a little bit on that topic, and I've found that there's enough validated scientific concern to make that not something we should do without further research. So in terms of increasing the number or the amount of oxygen produced by photosynthetic plants and microorganisms, cyanobacteria, I would say, in my mind, the best is to preserve some of the land habitats that we are currently devastating and have devastated in the past in terms of their photosynthetic biomass. We right now, I mean, previous environments have been degraded. Right now, Brazil is undergoing a lot of massive removal of their jungles, and that's releasing a lot of carbon and reducing carbon capture capacity as well as oxygen production. I think some of my colleagues would have some, it's a very interesting question. Thank you for that. And maybe some of my colleagues have some answers as well. Well, we've had a
Cyanobacteria have more than 50% of proteins.
Yeah.
So they are a good source of proteins as
We
well.
can start multitasking.
Yes,
in a way.
yes, and they can be used as fertilizers for agriculture in space stations.
Okay. So we are back.
Interesting organisms.
Sorry for this technical problems. We are back, and I would like to ask the colleague Joe Pollock if he's online, because there was a question for him. Oh, Joe, good morning.
I'm here.
You are muted.
Oh, no, I shouldn't be. Can you hear me?
Okay, yes, yes.
Okay.
So Laura Lieto yesterday had a question for you, so you can answer today. And so I will give the floor to Laura.
Hi, Joe.
Hi, Laura.
Hi. I have a question. That was a really interesting presentation, your presentation yesterday. I think that I understood a few things about butterflies and caterpillars and fish eating sponges on the reef. That was really wonderful. One of
It
the
was a
things
little
that-
rushed. I'm not used to speaking in 30-minute increments. I need at least an hour, it seems.
Well, I mean, please have mercy for my really limited understanding of your research, because I'm not a scientist. I'm an urban planner, so
Right.
I'm just the the new kid on the block. Let's say that. No, I was really curious about this story of the turtles, because I mean, as I understood, it's like, in order to corroborate your research and your finding, you went back to historical sources of ships crossing the ocean between Europe and the Caribbean. And at that time, as I understood what you said in your presentation, it was like you found out that turtles ate sponges with secondary metabolites at that time. So that's why basically the Portuguese and the Spanish didn't eat the turtles. I'm just joking. But my point
Oh no,
is,
you got it dead on. That's exactly right. Yeah.
okay, so my question is, how often you resort to other sources of knowledge that could be history, the history of navigation in this case, to get data or information that corroborates or question your work? Because that was really interesting to me as an example of hybridization of what we non-scientists understand your work of scientists, that is really precise, data-driven, very solid, very robust. And so this very, I mean, very humanity-oriented idea of resorting to data or stories from history, that was really interesting and fascinating to me. Thank you, Joe.
Sure. Yeah. I wasn't able to quite give you all the details on that part of the story, which I agree is one of the more interesting aspects of where this research has gone. The historical ecologists who came up with the estimate for the total number of hawksbill turtles that may have been in the Caribbean at the time that Columbus and Europeans made it over to the New World, that is a different group. And one of the people that have worked with that group is Jeremy Jackson, who's a very famous coral reef ecologist. But some of the students that have worked with him, and others as well, actually specialize in combing through captains' logs in the British Museum and in other locations in order to get the data that they then use to try to estimate populations based on records of nesting. Also, they use those turtles for food, mostly green turtles for food on ships, because they could turn them upside down and stack them like cordwood in the hull of the ship. And they would keep for months and months. So they had fresh meat. Of course, it was terrible for the turtles, but as far as the sailors were concerned, this essentially allowed them to go great distances, which they couldn't otherwise do. So that's where the estimates come from. And the 11 million estimate is, of course, an estimate. I mean, it's they can't possibly know that. But where it gets interesting is that if you look at the numbers that are in the Caribbean now, for every one hawksbill turtle that you see today, there were 400 if you use that estimate back in at the time that Columbus made it over. So the amount of grazing pressure that these large vertebrates were the amount of grazing that they were doing on the population of sponges on the reefs. And by the way, where Jeremy Jackson got involved in this is that green turtles were doing the same thing on seagrass beds. So the estimates of green turtles, which are primarily herbivorous, were huge, and they were mowing down Florida Bay on a regular basis. And we have no concept of that now because they're long gone. Anyway, those estimates are one thing, but then this really interesting data set about how nobody ate hawksbill turtles then, but they do now. And we know that the hawksbill turtles on the reefs now are only choosing palatable species. And so it's very easy to put 2 and 2 together to see that, of course, when you have 400 times the number of turtles on the reef, they're going to graze down those palatable sponges very quickly. And then all they're left with are the ones that they prefer not to eat, that perhaps taste, not as good to them. And the result is going to be their own meat winds up tasting bad because they're ingesting a lot of sponges with strange secondary metabolites. So yes, that's now you're talking about the integral nature of looking at data from other disciplines. And interestingly, after we published this story, a paper crossed my desk from a paleontologist who was studying glass spicules in sediment cores in Panama. And they saw this sudden sharp change in the kinds of spicules that were in those sediment cores that were taken on reefs that exactly come at the time when European explorers reached Panama. And what it shows is that the sponge populations started changing radically at that time. And and the species that suddenly appear are the palatable sponges. So the people that were doing this study had no idea about our work on chemical ecology of sponges. Fortunately, I was able to contact them, and it made their story much more interesting because they then were alerted to the work that we were doing. And it all made a much more sense because this was a bunch of Polish researchers who work on sponge spicules, mostly on ancient reefs in the center of Europe. And they had been doing coring in Panama and saw this dramatic change in spicular abundance in their cores. So all of that seems to continue to come together from disparate sources of data.
Thank you so much, Joe. Now you opened a new window for me. Send me the paper over.
Sure, I will. I've got it.
I'll try to figure out something, but yeah. It's incredibly interesting. Thank you so much. Thank you.
My pleasure.
So Joe will be in Naples next May, so maybe you can continue this interesting discussion in presence and maybe eating a very nice lunch or dinner, pizza. Okay, thanks, Joe.
Sure.
Now the next participant. Hi, Valeria.
Hi, do you
We
hear
cannot-
me?
We cannot hear, but we cannot see you.
Okay.
You need to switch on the camera, probably.
So.
Okay. If you can't, just start to presenting, introducing yourself. Where are you in this moment?
Can you hear me?
Yes.
Okay. So, hi everyone. I'm Valeria Berbetti. Actually, I'm a PhD student at the Department of Biology of the University of Naples, Federico II. The main topic in my work is comparative endocrinology. And today I have a question for Professor Gerwig. So, in relation to climate change, in which way the increase of temperature can alter the growth of bacterial community in marine ecosystems?
Did you get the
I
question?
couldn't hear that.
Can you repeat, because we cannot hear really clear? So maybe
Okay.
just a bit slower, Valeria. So you
Okay,
are
so
from the
in
Department of
relation to climate
Biology.
change-
Did I understood well? You are from the Department of Biology. Oh, we lost. Okay. Valeria, can you hear us? Okay. So I was saying you are from the Department of Biology.
Yes, I am.
Okay. Okay. So can you repeat your question for Professor Gerwig, just to
Yes,
be
I want to
slower?
know if there was a relation between climate change, the increase of the temperature, and the growth of bacterial communities in marine ecosystems.
Climate change and
Increasing
microorganisms.
temperature and
Yeah.
something else.
And increased number of bacteria.
Bacteria and marines.
Yes.
Aha, okay. Well, thanks for that question. Yes, in general, increased temperatures to a certain level do lead to metabolic conditions that favor microbial growth, but
Okay.
not of all microbes. Some microbes are psychotropic, that they really have optimal growth at very cold temperatures. But I would say in general, I would expect higher temperatures would lead to increases in microbial growth. Can you share a little bit about your interest in that question and where that might lead you?
Valeria,
Okay,
what?
so-
Yeah.
No, tell me. Sorry.
No. I was just saying if you did, you get what Professor Gerwig was selling. So tell us more about your research, your background.
Yes, I studied the effect of climate change on the reproductive health status in recognized bioindicators, mainly vertebrate and invertebrate. Climate change, such as temperature variation, pH, and salinity alteration of marine and terrestrial ecosystem. So this is why I made you this question, because I was very interesting in relation to climate change and growth. So
Yes. So do the students have the set of slides that I had sent previously?
Okay. Germana, ci hanno le slide? We can send.
Okay. So Valeria, in that set of slides, you'll see a slide that I
Yes.
discussed briefly with the reference there, which is actually quite a nice reference to the results of climate change causing an increase in pathogens of vertebrate and invertebrate marine species in particular.
Okay. Yes.
And
Yes,
so that reference, I think, will be very helpful to you.
Okay. Thank you.
Thank you to you. Thanks for joining this workshop and for your questions.
Thank you.
Next. Hi, Luca. Luca is working in our lab. It's not coming in New York. So, hi, Luca. How are you?
I'm fine. I'm fine.
Okay, so
So I'm
it's a
Luca.
pleasure to see you online.
I can see you. I don't know why.
Oh, the streaming's not working.
Oh. Ma non ci vede, ma ci sente. Ci senti?
Yeah, yes.
Ah, noi ti sentiamo a bassa voce. Alza un po' il volume o mettiti più
Okay.
vicino al microfono.
Okay. Okay. Can you hear me?
Yes.
Hi, I'm Luca, and I'm a new PhD student from Federico II University of Naples, and I work with the Blue Chemistry Lab team with Prof. Costantino.
Alza il volume della tua voce, Luca, non ti sentiamo bene.
I don't know why.
No, just speak slowly, parla più lentamente.
Okay.
So we can get what you are saying.
Okay. I'm Luca. I'm a new PhD student from Federico II University of Naples, and I work with the Blue Chemistry Lab team. Our research focuses on discovery of new secondary metabolites produced by marine organisms and plants. My project will be on structural characterization of secondary metabolites from plants. And I have a question because my project, one of the challenge of my project will be reduce the impact of the use of the solvents. So my question is, there is a strategy can I implement to reduce the impact of the solvent use? I don't know. There is some solvent that I can use to make extraction from plants.
Okay. Luca would like some suggestion to reduce the use of organic solvents or maybe using some green solvents. Do you guys in US have some clues about because we still in Europe are allowed to use chloroform and dichloromethane, but we also in Okay. Luca, are you there? Non vediamo Luca. Okay.
Okay.
Maurizio, you can give your. Luca, senti?
A little bit.
No.
I can see clearly.
Can you hear what we're saying?
Not clearly.
The volume is low. What about now?
Yeah, now
You
I
can
see.
hear better?
Yeah.
Okay, I got. So your question is basically how to optimize extractions to reduce the use of organic solvents.
Yeah.
And you're working with plants?
Yeah, but we have not
Okay,
yet.
well, yeah, I can give you. Well, I got some experience, and I remember also that I switched instead of using organic solvents. I was working with microbial cultures. So you have liquid cultures, you have liters of cultures, and one of the options is to use solid phase extractions. And I was able to reduce the use of ethyl acetate, hexane, or even butanol, to just switch to get everything concentrated into the solid phase extraction cartridges. So you have several options, and then you recover it with mixtures of methanol water. Now, that would be one adaptation that you can make into the plant tissues when you can, because there are several strategies, and you can lyophilize the plant tissues and then use some mixtures of with several polarities, mixing with water and some organic solvents like methanol, and then just get into the proportions, or try to get everything. I think it has to be tested, but in theory, one of the universal solvents, like every single cell has water, right? So all the molecules have certain solubility in water. So if you can trap everything into a solid phase extraction, you have a C18, you have C8, you have BHL, you have many, as you see in dissolved organic matter from, let's say, seawater. You have also solid phase extraction. You concentrate everything into the cartridges and then just elute them for your liquid chromatography or your fractionation or whatever. Then you are not using that much of any organic solvent like dichloromethane or chloroform or anything else. But you should check into the plant science metabolomics approaches. I can look for some papers, and then you can send me an email and I can give you some ideas. I remember Justin van der Hoef a few years ago was working in kind of metabolomics approaches. You have some also in plants. Lyophilizing is a good way, and then just do an exhaustive extraction. But yeah, I mean, you have to test, and that's something that I can think of. And the reason is because then, since we're working, maybe Bill will suggest something as well. When you work with algae, which at the end you can have a similar approach to plants, terrestrial plants, and just try to get a similar approach. In algae, you have also exposure to seawater, so you need to tackle those challenges in how complex your mixtures are, and try to reduce those organic solvents. So I think that the solid phase extraction resins like XAD, when you try to get everything in the liquid form, and then just use those resins to concentrate, and you can use that resin or those cartridges also a fractionation step. So it's also a cleaning up for your liquid chromatography, but it can be also for purification purposes as well, prefractionation steps. And that's an option. Sure.
Thank you for your answer. Thank you.
Yeah. Maybe just to share another thought on that. This is a new issue for us that's come down in the US and NIH, indicating that methylene chloride is no longer going to be an acceptable solvent to use. Methylene chloride was our substitution for chloroform. And the mixture of a halogenated organic solvent like chloroform methylene chloride along with methanol is the standard extraction protocol called Bly-and-Dyer extraction, which we've been using for more than 50 years. So it's this is calling for a major change in the way that we do extraction technology in an academic lab. Ethyl acetate has some properties similar to those organic solvents, but it's not perfect. It isn't as efficient in extraction. And I think we're going to need to shift to new technologies like supercritical CO2 extractions. CO2 is a fairly non-polar, and then with co-solvents like ethanol or methanol, one can get quite efficient extractions. We're not doing that in my lab at this time, but I think we need to start to think about moving that direction.
Definitely. Yeah. So definitely, I agree with Bill. We should go in this direction. And also this is, thanks for the question, Luca, because this go in the direction of this workshop, because of course, climate change is also related to the amount of solvents and organic solvent that we have been using in the past. I remember that I was a postdoc student and I came in a lab, in US lab for my first time at that time. So some years ago, already we're not using chloroform. So many years ago. And we still use some chloroform in Europe. So definitely we need to work in this direction. And this is something that we need to address in past, very few years, I guess, because otherwise we will improve this situation that, of course, is not going well. So thank you, Luca. See you soon next week.
See you soon.
Bye.
Bye.
Ok, sì, Annarita, dai la parola. Ok, Annarita, can please. Annarita is a former
Can you hear
student.
me?
Yeah. Hi, Annarita. Nice
Hello,
to meet you.
everyone.
Hi. She was a former PhD student in the Department of Pharmacology of our university, and now she is a professor, right? At the
Yeah, I'm an associate professor, in pathology. Yeah.
Okay, so please give your point of view, and thanks for joining us.
So I am an associate professor in pathology at Weill Cornell. I am a vascular biologist. My lab, what we do here, actually, we are interested in studying a class of lipids called the sphingolipids in cardiovascular disease. And I've been at Cornell for 13 years, and I first came to the States for my postdoctoral training, like many of you, I guess. And then eventually I stayed, and I got a transition grant from American Heart Association, and I started my lab at Cornell. And I've been here since 2011. And I am not sure, actually, Valeria, if you give me some...
yeah, we're talking about climate change. Yeah, we are talking about the climate change and the impact of our society, also the impact on the research. So,
well, this is a very important topic and a very sensitive one. It's very obvious that there have been dramatic changes. You know, in the last 2 years, for instance, We haven't seen much snow in New York. The winter is becoming a little bit snowless. And I think this is part of this also changes. While in Italy, we have seen an increase in temperature incredible in the summertime. And, since I was a kid in the last, what can I say, last 30 years, there's been huge changes in the temperature. As many of you, I'm saying here maybe something that many of you have already mentioned and noticed I'm stating the obvious. And I think we need to become more sensitive to this issue and also become more educated about about the issue per se and what can we do actually to try to have an impact on that. And perhaps I'm going to learn from you from this conversation today, since I also need to educate myself more on the topic. We are scientists, but it's a different field, right, that we are focusing on. And definitely we are very interested and sensitive to this topic. And perhaps we can learn from you today, too.
Thank you, Annalita. Maybe you can give us some suggestion to the participant how to start a career in US, also to our Italian participant. What was your experience?
Yes. So I think, first of all, don't be scared to go behind the Italian border. There is the whole world out there and can be quite beautiful, exciting experience that is going to change you, not only for professional point of view, but is going to change also from your personal point of view. And don't be afraid to do that, one. Second of all, find what you're passionate about without thinking too much of changing and getting out of your comfort zone. Because if you follow your passions, then you will never work a day in your life. And I think that's the best suggestion that I can give. I didn't know that when I left Italy and I came here, I literally followed my passions. I didn't even know if I was going to be able to start and have my own lab. And eventually things turned out good, exciting. And I had other students from Italy in my lab, many of them from Europe, actually. And some of them, they stayed. They didn't want to go back to Italy, but not because this is a negative thing, actually. It's not. But some of them, they came here at the beginning with the thoughts to definitely go back as soon as they could. They start to see the world perhaps from a different perspective, and they start to find other things that they might be interested and passionate in pursuing in life. One Italian, for instance, a postdoc that came from University of Trieste. She did the postdoc in my lab, and then she continued in a biotech company. And she's very happy. She still is in New York, and others, they continue and stayed in academia. So I think the postdoctoral experience or the PhD abroad is an opportunity for you to understand where is your place in this world. And I think you should embrace it at the first occasion. Yeah.
Thank you. So, we are in presence here in this beautiful location of the University of Naples, in the Roosevelt Island. It's an incredible place. We have an incredible view from here. So it's probably in the future we can try to do something together, some meeting here together, also with the lab of our colleagues, pharmacology colleagues in Naples, and take the chance to work together in this beautiful location. So thank you, and maybe we can catch in the afternoon for a coffee. Okay.
Sure. Looking forward.
Thank you. Bye bye.
Bye bye.
Okay. Mohammed, are you there? Oh, okay. Mohammed. I don't know my pronunciation.
Good morning, everybody.
Hi.
Hello. Good
Hello?
morning. There. Can you hear me?
Yes, we can hear you. Hi, let's introduce yourself. Thank you.
I am Mohammad Tosif, and I am first year PhD student at University of Basilicata, Potenza. Basically, I am from Pakistan, and I started my PhD in the field of the applied biological sciences at the University of Basilicata. So basically, my research project would be the agro-food waste valorization. There has been already a discussion from the participants today. Like if you asked questions about the extraction, and I would be working on the plant matrices, and my major project would be the optimization of the extraction, and by using some green solvents and considering the main focus, considering focusing on the circular economy principle. So this is a kind of brief introduction of my research project, which I am going to start soon.
Do you have a question for us?
Some of my questions have already been answered. Like recently, the last question asked by Luca, that was also some of the problems I consider, every students consider while working on the extraction, like how to and what to look as a green solvent, a green circular economy, environmental sustainability. Because my project is on the food waste, agro food waste valorization. So if we are considering at one point to solve the problem of the environment by utilizing the waste and bringing it back to the loop of the circular economy, then by working on this topic, particularly by extraction on the waste, there becomes more responsibility that how, while we are talking about the circular economy and the environmental sustainability, and then what we are doing during the process. So there are some points or some kind of questions in my mind, like the solvent, which has been answered already. So I would like to ask or add an other point, like there is another approach like deep eutectic solvents. So I will be trying that bio-based solvents. It could be a combination of organic acids, some sugars, or some amino acids. Can you hear me?
Please. So we understood that you had the question about the solvent. It's nice that there is this awareness of this problem of using green solvents, and you got the answer before. And do you have another question?
So I would like to ask about some like NADS, natural deep eutectic solvent. This is one of the growing approach these days. I think a combination of organic acids and amino acids or sugars. By using this combination and doing the solid phase extraction. So I would like to from anybody any suggestion or comments or the trends in the natural deep eutectic solvents. So this is one question, and the other question is uh, like in a general context, in your opinion, it could not be a concrete question, but where we can see ourselves, our research in the future, that while struggling with the solvents and adoption of the new technologies at the lab scale, like professor mentioned about the supercritical fluid extraction. So these days, microwave assisted extraction techniques and ultrasound assisted extraction techniques are quite popular at lab scale, but not the supercritical fluid extraction. So in this context, my question is how far we can see all these things combined as a one solution in the world. I mean, at an industrial scale, how we can scale up these, are in how much time, or how far you people see when these technologies or these thoughts which are at lab scale, that we want to do everything perfect in sustainability, we want to achieve in the circular economy, and how we can, or how far you people see that these approaches will be in the market, or it can be adopted globally or at a larger scale. So, and your opinion on this, or your thoughts on this scalability, basically.
I'm sure that you have an answer about the scalability, scaling up extraction if we have this problem of reducing the solvents.
Mohammed, I can give you some talks, but I just wanted to make sure if I understood well what you're working on. You're studying or investigating the waste of what? Agricultural waste? That's your project about?
Yes, food industry byproducts are residues one of the most popular grapes, grape pomace, olive pomace. Are I would be working on the bean spores, any kind of plant-based industrial waste coming as a residue or byproduct.
Because two approaches that I see, because it's really hard to see sometimes is in how long we are going to change the practices. As Bill was saying, I did my PhD in Brazil, and I saw, for instance, people in the marine and natural products were still using those chlorine derivative solvents, while others were not necessarily focused on that. And the reason is, my thinking is the two approaches. One, that if you use sensitive techniques on whatever biological samples you are using, you are working in ways that is probably more complex as well. If you use sensitive techniques to detect what chemical classes are present in your biological sample, then you focus or you try to target which ones are associated to your research questions. Then you can optimize the extraction for those. And the other is the more holistic approach, is that you want to capture the whole, let's say, metabolome. You want to see everything. Then you need to do a more global approach, and then just try to use kind of strategies that cover most of the chemical classes that you can find in those. Like chemical classification is also another thing that probably you will discuss, but you will learn during the process, and what kind of molecules you are targeting, or you are detecting. Now, if you focus on one molecule, then it's kind of easy, because if you know what you're looking for, then you can adjust, okay, what solvent you use, what pH you use, etc., etc. Now, if you want to capture everything, then you use more comprehensive approaches. There's more wide range of polarities in solvents. And it also depends on the amount or the volume of your sample, how it's a small or large scale of the samples that you are using for your approach. But yeah, I think that for now you can probably follow up by email, and specifically what your question would be as far as you are testing. Because just to tell you something about, and I've seen that in different projects from different students around the world, is that something that has not been fully tested is what is the stability of a biological sample before and after it has been prepared for whatever approach you want. You use LCMS or LCUV or LCN like scattering approaches. The more unstable your sample is, let's say in solution, of course the amount of molecules that you will end analyzing will be wide larger, because you will see the molecules that were originally in the biological sample plus the degradation products of those during the preservation time that you have. So at the end, it depends on what you're using and what is the research question that you're asking for.
Okay, thank you, Mohamed. Have you another question?
not question, but I would like to. Basically, this is not a question that how and when and in which time we will achieve the sustainable scalability of all these extractions or these natural products and metabolites. But in your thought, in everyone can pass me a remark that what do you people think that is there our society outside of the laboratories is responsible to adopt these technologies by sacrificing their economics and any comment on this, that what do you people think that how our whole society will be adopting all these environmental sustainable or circular economy principles, or are we ready as a global nation to adopt all these technologies, and how it will shape our society economically, or what would be the resistances we will face from the economic point of view by adopting new technologies. It may not be economically feasible. So as a global nation, uh, what do you think that how much we are ready to act on all these sustainability principles? Your thoughts, or anyone if want to comments, that is the only thing from me.
Okay, Bill.
So, Mohammed, sorry, it's a little hard to hear, and so we may not have heard your question quite correctly. But as I'm hearing it, you're asking the general question of, is society ready to adopt these new methods that are going to be more sustainable for the future, and what kind of timeline is involved there? And Laura, you may have some comments to make about that as well. In my brief experience on this planet, it seems that certain things change in society as a result of market forces, and then other things need to be changed by legislation. Sort of when we have a longer-term objective or need, those things don't respond necessarily to market forces, but more to a government saying, we must change the way we make extractions. And that legislation is coming down in the United States, and as I understand it, in Europe as well. And so we will simply have to change. We will simply have to use new methods for extraction of our samples. There are already methods available, like supercritical CO2 extraction, but I'm sure there's other methods we haven't started to think about. And we will think about, because we will have to think about, and we will have to make those studies in the future. So I think society is ready to do this, and I think the legislators are going to bring this about, and we will have to make these changes. But anybody else?
Okay. Can I make a more general comment on that? Mohammed, I'm not a chemist, I'm not a biologist, I'm none of that. So I'm just, but I'm interested in this conversation. And it's in a larger picture, all of the climate change debate, it's like we actually experiencing a separation between science and politics, which is really, really negative for the outcomes, for the future of the planet, whatever, next generations and things like that. I think that science as a whole should be more vocal about, some arguments. For example, you were discussing before in terms of new drugs discovery through improving biodiversity from waste, right? And as I understood that conversation before, it was like, okay, there's no such a thing as waste as something that we gotta get rid of, because waste can be waste for some organisms and can be a resource for other organisms. Or okay, I understand this, there's complexity involved, and that's a good point. But how you as a community act in front of larger scale waste production, because that could be, you know, an issue that you could contribute pretty much in terms of recycling, in terms of many things. And I know that many policies and many techniques are relying heavily upon the scientific discourse, but I think that if we engage in a more compelling political conversation and bringing the contribution of science to larger social problems, that could be really interesting. There's a beautiful article. It came out on the New York Times a few years ago, and it explains pretty clearly how, at a certain point in history, scientific discourse was central in any political decision about climate change. And then after some point, that was just falling apart, at least in the US. And I think that is a really interesting point to understand. Sorry, Mohammed, maybe I just took another route, but it's really interesting to listen to you guys. Thank you.
Thank you, Laura, for your point of view. And we can thank you, Ahmed. Mehmet, we can continue with the...
Thank you so much.
Hi, Silvia.
Hi everybody. Can you hear me?
Yes.
Okay, thank you so much. So good evening to everybody. It's always a pleasure to be here. Probably it's my third summer school on nature products, and it is always really a pleasure to be here. And I want to thank everybody for the talks of yesterday that were really inspiring and interesting. So really, they gave another point of view, and it was really, really interesting. So I just want to introduce myself briefly. I am Silvia Scarpato, and I am a postdoctoral researcher at the Stazione Zoologica Anton Dohrn in Naples. So I call from Italy. And my research activity is focused on the analysis of complex extracts from marine organisms. So ranging from microorganisms, sponges, corals, and so on. And I use a combined strategy, and then targeted strategy, actually, combining LC-MS MS data and molecular networking, trying to identify, detect, and isolate, of course, and fully elucidate new bioactive nature products. Actually, I have a question probably for Mauricio, but of course, everybody can answer to that question. So I think that the potential of MASTI, of your tool and your project, the one that you presented at the end of your talk of yesterday, probably it is called MS Collaboratory, but I don't know if I'm wrong. I think they are really impressive, because in this way you can just collect the data of everybody, trying to reduce time in I think is actually in the replication of known natural products in the extract, if I understood correctly. But probably, in my opinion, I think that the major issue in natural product research is to elucidate the structure of new unknown compounds. So, I mean, with the dereplication, you know that in some cluster you can identify some nodes, so some compounds, and you can understand, or trying to understand using the mass fragmentation, some analogs that are connected to the one that you know. But if you have some cluster in which nothing is identified, and even if you use a larger database, you cannot identify these molecules. Is there a tool or something that can help you to recognize some substructure, or at least to categorize this molecule, trying to understand into which chemical class it can belong to, or at least have some idea of the substructure? Because, you know, sometimes you don't have the access to NMR or some other instruments. So I don't know if you can answer to that question. So this could be really helpful in our research. Thank you so much.
Please, Mauricio.
Yeah, Silvia, you hear me, right?
Yeah.
You can hear me. Okay. Okay. Thanks. Definitely, really good to know that you're using the approaches. There are at the Dresden lab, we work with several projects. We collaborate with too many people. So you will see a mixture of tools and workflows that are used for many reasons. And it's good that you at least have like a specific question, and that is an interesting one. Because what if we don't have any identification, even in a molecular family? That's something that I show a little bit, but you can use these masked tools to see where you can find the molecule that is present in your data set, and to see if it's present in any other, to give you some insights. But an interesting one is the one that you ask is, what if there are some tools that enable us to use the same approach of comparing or doing the spectral alignment, but at the substructure level?
Yeah.
And yes, there it is. We are working in some that is probably in the near months. In one of the projects that we're working on in the computational side, as led by one of our postdocs, Shipei, is working in something called reverse cosine, when basically you imagine a molecule, you don't have the structure. And in natural products, as is our field of is very familiar with, we have several moieties that, let's say a polyketide, just to give you an example, a polyketide, you have an anthraquinone that is decorated with several sugars. It can be glycosylation on the oxygen part, but also in one of the carbons, it can have the glycosylation. But you don't know anything about this molecule. But if you are able to detect the fragments of the sugars, then you know at least part of the structure.
Yeah.
And with the reverse cosine, we are able to get this similarity score to substructures. And that in this specific example, of course, I don't have the slides, but you have to imagine, some of the people here are imagining the anthraquinone, and then just imagine these aromatic rings that is very strong. You cannot fragment it very well, but you can get the fragments of the sugars, because then you see the fragments. So you have 120 Dalton's neutral losses, and then you see them. Now, if you apply strong energy into the instrument to be able to fragment, then you will be able to fragment that anthraquinone, but it will be like kind of an explosion. So you will see tiny fragments that might not meaningful to be able to do a spectral library, because no one else has deposited such a reference spectrum. However, at least knowing that it has those sugars, you will change your focus on extraction and say, okay, I need to probably go into the more polar side and then try to get more of this molecule. If I don't have this fragmentation spectra in any other data set in the whole data set, depository, mass spec repository, then at least it helps you. Knowing that there is not a single fragmentation spectra in the entire repository, at least it's good news. And then you focus that. So one of the strategies is that reverse cosine. There is also a few, well, a couple of years ago, there was Justin van der Hoef. They work and also implemented into the GMPS infrastructure together with Ming the mass2ML approach, which is at the substructure level. So you are able to annotate fragments similar to the neutral losses. You have a neutral loss that you observe, then you know at least that there are some sugar moiety there, and then you are able to annotate at the substructure level. Then you have an acetylation, you have some of these losses that you can annotate, so you can use a library of substructures and then try to get some insights into this. So at the end, the conclusion is some information is better than nothing. Just knowing
Yeah,
that the molecule that you have in your dataset is also present in a microbial dataset, it's also information. It's useful. So sorry to take the long answer because there's many tools that we're working on, but that's why we work with collaborators like you that are interested in something that is not available. We just put the efforts to work on this and then probably develop it because it's needed. Thanks.
thank you so much. Thank you.
Yeah, and Bill.
Yeah, so Mauricio gave you a wonderful answer. And just to build on that example of an anthraquinone with some sugars, and by mass spectrometry, you see the cleavage of the sugar units, and so you get some clues to your molecule. Don't forget some other data points. Typically on LCMS, you also have a diode array detector for UV absorbance.
Yeah.
And that UV absorbance can also give you valuable clues as to what type of structure or structural features you have in your molecule. In that example, the anthraquinone would show up as a distinctive UV chromophore, and there are UV databases that you could compare to and get some further clues as to your structure. So that was one point I wanted to make. A second point is, in those cases where you do have enough molecule that you can isolate it as a chemical substance in a milligram or more quantity, you can run NMR in your laboratory or perhaps in another laboratory. Or send the sample to us. We can run it for you in San Diego. But a technique that we've been working on for a few years now is called small molecule accurate recognition technology, or SMART. And it uses HSQC data, that's proton C13 data, and compares it to a database of about 150,000 pure natural products. And it's been remarkably accurate at showing you if the molecule is in the database, it will very nicely dereplicate that for you, show you, okay, this is a known molecule and here it is. Or if it's related to a known molecule, to some degree, it can show you that it has some related structural features. And that's a big clue in terms of assigning data, as you say. Well, because, yeah, the case I wanted to also share, the way we express the case that you brought up, it's an unknown unknown. You don't know what kind of a molecule you're dealing with. And these clues that you get from the GNPS, from UV data, from the SMART tool, can tell you what kind of molecule you're dealing with, and then you can compare it with data sets in the literature and accelerate the process of figuring out what it is. I hope that's helpful.
Yeah, it is. Thank you so much.
Thank you, Silvia, for your questions. And we have another question.
Good morning, everybody. Could you hear me?
Yes.
Yeah. I'm Vittorio Carlucci. First of all, I'd like to thank all the organizers and all the speakers of this international summer school. And I'm PhD student at University of Basilicata, but currently I'm doing my PhD abroad. I'm in Prague, so I'm working at University of Chemistry and Technology in Prague. In general, I'm working with natural products and their biological activity. My question is about the GNPS. I started to use GNPS in my research, and I had some issues because I'm at the beginning, but I would like to ask you some advice for this platform. I'm working with untargeted analysis on different extracts from plant species, and I would like to do the untargeted analysis of to investigate the secondary metabolites particularly, I'm interested on specific compounds such as polyphenols. And I, before starting the project on GNPS, I used different software such as MS Dial and MS Cleaner and MS Finder. And my question is, could we upload the raw data or clean data on GNPS? And which is the best way to get good results for the identification from by GNPS? This is my question.
Well, Vittorio, so you are in Prague, right? Czech Republic. You can hear me, right? Okay, so you're in Prague and Czech Republic, right? So you have nearby to your lab, I think, Tomáš Pluškal, right? Tomáš
Yeah, I know.
Pluškal. Yeah, so you know, you got the MC-Mind team there as well. So you have a lot of experts also in MC-Mind, but also in GNPS. So they, you will get help very quickly there as well. So for your question, when you say clean data, that might have different interpretations. For the GNPS as infrastructure, because then you can imagine as an infrastructure, you can imagine GNPS as an umbrella of several workflows and tools. And a starting point is to have the data into the public repository, which is massive. And then the basic things that or the fundamental things that we ask for is for the researchers like you to upload the raw data and the MCML files of the raw data. And both of them, we treat them as raw data. The reason is the raw data directly from the instrument is the original one. So it's possible that the softwares get better and better, and in the future, we are able to grab the whole information that is deposited in every single file, because it's more than the peaks list that we have and we use. And that's one thing. Now, the MCML, you can imagine as a standard format in the mass spectrometer community. Then you have the peak list. So basically, you have the precursor ion and the fragments with the intensities. That's the information that we use for the network approaches. Now, you work with those, let's say raw data or MCML data, and you use other softwares like MC-Mind or MS Dial to get kind of a processed files, which is the list of the peaks with the intensities, peak areas, and that can be, at this point, a clean data. However, you do some extra downstream processes. Let's say in your experiments, you have blanks of analysis, extraction blanks, analytical, quality control, pools, quality controls, etc. for your metabolomics experiments. And then you might include the information from those files as well, and then remove blanks, etc. So all these processes at the end to have a table that is used for your statistical analysis, it's another called clean data. And it's something that only you process the data, only you know. But then if you associate all the files and everything into the data set, then provide the information that what each of these files contains, then you have clean data plus information that also the community can use. But you already did that, and you have already several steps of clean data. So with that, probably I answered your question, but then you have several tools that you can apply for the specific question that you have. But yes, you can provide as many files as you want, but importantly, the raw and the MCML files are the most important. And the reason is because in the future, we can mine the data. In 10 years, we can mine the data. So even that you process the data in one way, nothing is perfect, but we can always go back to the original files and process everything again. And nothing will change because the data is already deposited. I hope you got the whole answer. And Vittorio, yeah, feel free to reach the Pluscal team. They know us. So you probably talk with Robin, with Corina, with Tito, and many others. Try to interact because it's something that we do and that facilitates the research. Just communicate with many other team members from around the world and feel free to reach out as well.
Thank you so much, all of you.
Thank you, Vittorio. Vittorio was one of our former students in the last edition of summer school in Maratea, and it's nice to see that you are grew up in your research. It's a pleasure to see you, and thanks for joining. And so I would like to ask to Joe if he has any other comment or just to tell us bye-bye before closing the session.
Bye-bye. Thank you all for inviting me. And and I'm sorry I didn't get to talk to all of these people I know. So, but you know, that's the thing about distance Zoom meetings. So, anyway, I hope you're having a great time in New York, and I know everyone's having a great time in Italy. So,
Joe, see you on May.
okay,
Bye.
looking forward to it. Yeah.
Okay.
Okay. Signing
Bye.
off. Bye-bye.
Have a nice day. Thanks for coming. And also, there are some colleagues in connected with us. One from the Department of Pharmacology, strictly connected with the Anna Rita De Lorenzo. I don't know if Maria Rosaria would like to share your thought with us if you are there, and if you can. Hi.
Hi, hello everybody. So
Actually,
thank you. Tell me.
Actually, she should come with us here, and she can't at the end. So thanks for joining us in a virtual mode. Please.
Thank you all for this interesting international blood summer school. And of course, I'm not expert in this field, but it is very interesting for me to listen and to learn something new for me, because I'm a pharmacologist, but in a way it is always important for us, for new drugs, especially for cardiovascular system, that is my topic, to understand, to see new and different point of view. So thank you for inviting me. Thanks.
Thank you, Maria Rosaria. And so you see, we have this wonderful location here in Roosevelt Island. So we have to take the chance to do something here together with Anna Rita in Cornell. So thank you.
Yes.
Bye-bye.
Bye-bye. Thank you.
Bye. So if we do not have any other comment or question, would like to do just 5 minutes of for a coffee, and then we will start the interesting workshop of Mauricio. Okay? So just 5 minutes, and we will be back for the final part of this session. That will be the workshop. This will be very interesting for you guys making all this question. Thank you.
Okay, so now we can start with Mauricio and his workshop, interactive workshop. So all students that are connected can use their laptop and can join. I can also write some question. If you have some question, you can write and we can answer.
Yeah, I'll try to finish quickly.
Yes.
Then in the 10 minutes, then we'll have at least see what questions they have, because the slides are available. They can follow up as a tutorial in their own time.
Okay,
Because
perfect.
that's why 30 minutes is really short.
Okay, perfect. So thank you.
Thank you, Valeria. So the first thing is that guarantee that they are here. They hear, right? Okay. Okay, well, most of them are in Italy. That's just buongiorno to everyone. Buongiorno to everyone. Okay. Okay. Let's start then, and then we have. I put it the QR here with the material. So in this link or in the QR, you can have access to the Google Doc, which has the link to the slides and also the link to the resources, which is the same Google Doc that has the link to every single tool. I will work on the MicroMass today and the molecular network, but then you have access to other tools. So you, depending on whatever you're working on, so you know that they exist. And I also put the links to the slide, to the papers that are related to some work that we have done. And some of them are basically the resources of each of the tools that we have. So we have molecular networking, feature-based molecular network, MAST, MicroMass, a recent one that was finally published last week, which is a work on with Daniel Petras and Madeline Ernst. They were working on feature-based molecular network stats, web and app. So it's kind of an educational tool to learn how to do some statistics with your feature-based molecular network. So the first thing as well, besides getting the material, is that I will give you an introduction of why we are talking about GMPS2, because then some people are new in using the platform, but then Ming-Wang, which is the developer of the GMPS as infrastructure, he moved to UC Riverside when he is now a professor. And then we are controlling and giving access to an account. So you are able to use the GMPS2. For this, I encourage you to sign the form and fill it up. Then I will receive some and I will distribute tokens later on. So you can access that. You don't need the access today, but once you start, you have the access to the slides, so you can just follow the steps, and then you can do it by yourself. Once I hear back from you, you got the form, I will send you the token, and then you will have access to the platform. And probably you are starting or in the middle or at the end of your PhD. It's good for you to know that these resources exist. Because it's possible that you are starting your PhD and then you have, in a couple of years, you have the questions like, hey, how can I analyze this? For instance, is one of the topics that I'm going to cover today is I want to know if the molecules that I'm detecting in my samples are actually microbial. How can I do that? So you know that the resources exist and how to access. The first step that I'm going to give is, and it's probably one of the questions that we have in the discussion today, is that I have my files that are public data. How can I actually analyze it? The first step is performing a classical molecular network, because then from that, you can use any other tool that is available and built on top of the classical molecular network. Here I will give you the overview of what we are going to do, and then you will have a complete understanding on when to start. So the GMPS data ecosystem, which is the one that I'm going to start now, and showing you what it actually means. Then we'll go for the networking approach, then the test data that I use for this one, then how to create the account, uploading the files when you have them, and then launching the molecular network. And finally, we will explore the results with some MicroMast. And let's just start with the GMPS data ecosystem. So the GMPS as it is, it has algorithms that are built, for instance, in the networking approach, which is the spectral alignment. I will explain a little bit more about this, but also the data repositories. So we put everything in the public repository. Is a massive located in the UCSD. So you publish your data, and then will be there forever. So you will not lose the data. You will have the DOE for your publications. Everybody will know that you have ownership on those data sets. And public data helps to build bioinformatic tools, computational approaches, accelerate the research with tools like the ones that I'm going to show you. And knowledge bases, that it's something that we are going to explain a little bit, is how do you associate information to every single data set and every single spectra that you find. If the molecule is from microbes, and the microbe was collected from soil or from marine environments, and where it is, etc. These all of the information that built into the knowledge base. Now, how is the interaction between the GMPS and GMPS2? First thing is that Ming Wang, which is the developer of the GMPS infrastructure, he is now professor at UC Riverside, and everything that is new, everything that is going to be built, like the mass tools that I will show you in a little bit, everything will be built on the GMPS2 environment and will be developed there. In the GMPS one, some of you, if you were users of this, you will not lose the data stay there, but anything that is new and we will develop will be only in the GMPS2. Here is just an examples of visualization tools. The reason is because with all the data that you acquire in a single experiment, just doing liquid chromatography tandem mass spectrometry, you generate thousands of spectra. So how you visualize those is a good way to inspect the data very quickly. So I provided the links here. I will not focus on this, but then you know that the GMPS dashboard is something that you can check raw data. If you see, for instance, you want to inspect if the peak that you were interested in, if you have a reference molecule that you were expecting to extract and you don't see that, then you quickly visualize if it's there or not. Metabolomics Resolver, I will show you a little bit on that, is that how you can generate a fragmentation spectrum in high quality and compare against others, and even use it to publish in your manuscripts as well. The Interactive Network Explorer, I will show you a little bit a screenshot of that, but it's basically you can visualize the network after you finish your molecular network. And for analysis tools, I will focus on the networking, which we are going to visualize, because we can see how many of the spectra are annotated. It means that they have reference spectrum, but also one of the MAST tools. The MAST tools is the single search spectrum, when you can find other data sets where your molecule is or has been detected. And the MassQL is one of the examples of the new tools. And then I'm not going to focus on this because it will be probably the time for another workshop, but you can think of the MassQL as a data mining approach. So if you want to look for a specific fragment of group of fragments that are characteristic of a molecule that you are working on, then you can see if you can find these patterns across your dataset or across the entire public dataset. So this is really good because it's something that you find one molecule, you discover a molecule, and you are pretty sure you got NMR, you got all this information, and you had the fragmentation patterns. You can see if, since the molecule is new, what other analogs might be of this one. And it's probably one of the questions that are at the beginning that there is another answer to this one. You can use that to find substructures as well. So it's basically, this is coming from SQL data mining that is using R. This is basically coding, and now it's getting easiest. Okay, so we gave an overview of the data, the ecosystem, and then let's go to the molecular network overview because it's the fundamentals, and some of you might not be familiar with this, so I just give a fundamental understanding on this. So you have your samples, biological samples, and we use liquid chromatography, tandem mass spectrometry because we generate several layers of information. One of them, as you can see in the plot here, is that from the liquid chromatography, we have a retention time, which is based on the interactions that they have with the chromatography. Then you have an intensity that is based on the ionization efficiency of every molecule that is detected into the mass spectrometer. And then you have also the mass to charge, M over Z value, which is the charge, the mass over charge, is the way how the molecules are detected and represented into the instrument. And additionally, as you can see in the plot here, is just an example on the stars that you see in some of the peaks are the ions that are fragmented. So you see to the right is that these molecules, intact molecules, are fragmented and then provide a fragmentation spectrum, which is characteristic for every molecule. And we use that to perform the molecular network approach and library annotations. Here it doesn't have the animation, we just make it fast. Is you have thousands of spectra here. It's really hard to analyze this manually. So if you can think of this as a tool of accelerating the prioritization of molecules that you are going to work with in the next steps. So here to give you a couple of examples is that we have these microbial molecules being well known in the natural products. They have strenotrypsin A and C, which are produced by Streptomyces strains. So you see how similar they are, and then they are only differentiating in the lipidic chain that is one of the decoration parts of the molecule. If you compare the fragmentation spectra, and then as you expect, there is a core that is exactly the same. So you see the core that is similar in both. So you expect that part of the fragmentation spectrum is exactly the same. So it's matching. And then part of the molecule that is changed, it has to be at the up range of the spectrum. So you see, okay, this is where the molecules are different. And this is what I mean when you have a substructure that is similar, and then you can find it. The metabolomics resolver is this, I provided also the link, is the way that you can compare using the USI, which I explained a little bit. The USI is the Universal Spectrum Identifier, so it's basically like a coding for every single spectra you have in any data set. So you can grab those two and compare them in a mirror plot. It helps definitively to get this analysis in a reproducible way. Now, to give you an overview of what molecular networking is. So you see here in this nice example of some tryptophan-derived molecules together with biotin. So you see that the tryptophan as it is intact, you have an M over Z value of 205, so you have the fragmentation spectra, Then you have some modification. You have an hydroxylation, then you have an acetylation, and then you have a replacement from nitrogen to an oxygen, and then you have an amide, and then you see the difference between these two is just basically 1 Dalton. So when you have the fragmentation spectra of each of them, then you can do the spectral alignment and then represent every single fragmentation spectra as a node, which is the visualization tool that we use for molecular networks. Then you are seeing how these spectra are similar based on the spectral alignment. The cosine score, which is the metric that we use to compare two spectra, considers the fragments, consider the intensities, and then vectorize those spectra, and then using the angle on the in the 3D space, then attribute an angle between two compared spectra, and then provides from 0, which would be two spectra that has no relationship between each other, versus 1, which would be exactly the same. So the closer to 1, it means that the more similar spectra are. The closer to 0, it means that they are not. Now you put in your thresholds and in your settings in the molecular network, for instance, something that is lower than 0.6, 0.7, it depends on you. I will show you later on where do you put this, will define if they are connected or not. So you see pretty evident that these biotin molecules are completely different to the tryptophan. So of course, makes sense that they form a different molecular family, and then they are not connected. And that's the fundamental of the molecular network. Now, to give you the examples, and then on the stenotricine, and here I just want to emphasize that these two molecules, which is also a new one here that was discovered using the GMPS approach a few years ago, is that you have just the difference into one of the amino acids. So if you put the Streptomyces rhodosporus strain, which is the green one here represented, then you may have several spectra, and one of them corresponds to the stenotricine C. Now, by analyzing the two extracts and building the molecular network of the entire extracts, you see the connection between one of the molecules that was discovered as well. We call it the stenotricine GMPS, because they have a difference of 41 Daltons, that at the end it was the difference between the serine and lysine. But if you wouldn't have run the analysis of both extracts together, we just have missed the connection between one and the other. And definitely this helped to annotate or identify the new one, because you already had the stenotricine C structure. So that's how it helps. The more data or the more samples or the more diverse samples you have in your studies, the more chances you have to have chemical coverage of your analysis. Now that you have the molecular network overview, you have an understanding on the fundamentals of this. I will just give you an example, one of the data sets, and you can do this for any public data set if you want to train yourself and learn the approaches. Is that from these data sets is one of the data sets that we are still analyzing. We have one of the first papers published a couple of years ago. And it's these samples that we got collected from the Waimea Super Transect project in Oahu, Hawaii, when we collected samples from plants, insects, water, and sediments. And then we want to investigate association between the microbiome and the metabolomes across the transects. And one of the interesting parts is that there are some parts of this forest that is at the top of the mountains that are reservations. So it's like almost zero human impact, which is very interesting from the our human impact on nature point of view. Okay, so whenever you have the chance, let's say next week, and you fill the form and you send me the interest in using the platform, or it can be later on as well. You got the token, you go to the GMPS2 and you do the login. You need to create the account. So you will create the account, then you will receive the token. This is where you place the token, because we want to guarantee that all the users of the GMPS2 are, well, real people, and then also from the academic institutions. And once we have this, then you can create your account. You log in into the account with your password, and then you are all set. Now, when you have your account, you can go to the file browsers. If it's the first time, of course, you need to start using it, right? So you can, similar to any file manager that you have Dropbox or Google Drive. You can also create here folders, and then you can link the data that you need. If you are going to create a folder, in this case for the workshop purposes, you can create a folder that is like workshop, and then you will have to upload the metadata that we are going to use for this one. I also provide the link to the metadata here. You can download it. It's a TSV file, and then you can upload it into your account later on, and it will be in the same format because we will use it for the analysis. Now, when we already did our file upload, we just go to the first window of the GMPS, the home page, and then you will look for launch workflows. And then you will look for the classical. Then it will be the classical molecular network. You can type to the right, or you can just browse, scroll down and find it. Then we just got the classical molecular network. We are going to provide a name to the job. This is going to be the same way as we are going to do for any other future job that you are having. The name that you are providing here is something that is meaningful to you, because at the end, once you will finish, you will see that the job will have an identifier, and will have the date, of course, so you will find it anytime. But sometimes you want to run a job and then clone it, and then change some settings, and then do it again. So that's why it's something that is more meaningful for you. So you can select the files that are going to use, and then for this, and this is something I wouldn't say critical, but it's important, because this is how you are going to learn how to search and use public data sets. So the same way I'm doing here, you can do for any other data set that have metadata associated as well into their data sets. So you have an identifier for the massive data set. So you can just expand the data sets, and then go to the massive and expand that one as well. And then you will look for this specific one. So you will look for the number that I provided here, the 85129, which is the Waimea Super Transect. You also expand it, and then you will see that there are two folders. One of them is the peak, and both of the folders contain exactly the same files, because it's something that in the data deposition of the original files, we created the MCML files. But also Massive, which is the repository, also was able to automatically create the Massive, the MCML files. So in this case, just go for the peak, then we'll have the files that we are going to use. And then you can collapse, because now we can add select the add selected file to input data file. So remember that we have selected the peak folder. Now you select it here, and then we'll go to your account and find the metadata where you put it. I put it also under the workshop folder. So I select again, check that. It means that it was selected, and then add it. Oh, and then add it to the metadata file. If you did that right, you will see, you can close this and you will see them selected. You will see that in the file selection, here it will provide the path to the file or to the folder containing all the files and also the metadata. Now, when you change the settings, and it's something that you can do, is you will go to the precursor ion tolerance, because we work with high resolution data, so you can be more strict into the settings, and it's basically is refining how many or how big the window for the precursor ion as well up to the fragments can be. So in this one, just change it to 002 and then keep the rest as default, and that is fine. I put it also the link here, and you can go to the table 1 of this paper published by the lab, written as a protocol, when you can also follow step by step. But it was in the GMPS 1, but it's the same fundamentals for the GMPS 2 that you are learning here. The most important here is that you will learn, if you are interested in, because it's something that is not obvious for everyone, what it means annotation. What is, when you talk about cluster index, which is basically the number that is identified for every single spectrum. And you will see, I will show you later on what that number is. It's just an identifier. Or what is cosine score, what is the replication, etc. So it's good terms that you need to learn or get familiar with. Then you will submit the job, and that's how it seems like it's easy, but then when you are doing it and you will understanding what you're doing, then it gets easier as well. So the job is running. One is, or even when it's running, you can clone it and then change the parameters. So it's like, okay, I don't want to get a cosine score of 0.7. I want to put it like lower because I want to cluster more spectrum. And I want to use, instead of 6 minimal peaks, I want to use 4. I would suggest that depending on the fragmentation spectrum, about 4 would be the minimum, because less than that, like what is the probability of having 2 or 3 similar fragments across the molecules? You probably will cluster everything, and that will be kind of difficult to see which are different chemicals, molecular families. In that, you can wait depending on how big the data set is, etc. You can wait a little bit more, an hour, or it can be minutes. Also depends on how busy the server is. But then here I'm providing the link of the job has already done, so you can also follow me in these ones as well, and that's fine. Once you have the job done, and it's something that I wanted to show you as well, is that you have the library match results. But in this case, just to emphasize as well, that you have this task ID, and you can see this number, and I will show you once we do the micromask where it's located. And also in the previous one that you have in the link to the job, you see that in the link actually is also the task. So it's basically the same. So you will, now you know where to find these numbers that you eventually will need, or you need to confirm if the job that you run is the same that you are analyzing. Because if you run many jobs of the same data set with different settings, you will figure out that at some point you get lost. But then that's the way you will see, okay, this is the one that I use with 4 fragments with a cosine score of 0.65, etc. And then you can click, and this is the first overview of the results, because for some, one of the key points in the analysis after acquiring LCM SMS data is I want to see how many molecules are known from my data sets. That's basically the one thing. If you didn't get any annotation at all, it can mean two things. Or either everything that you got in your sample is new, which I think is low probability, but it is possible. And the other is that probably the settings that you had were too strict, that not a single library match was captured with that. So that's the two key points. Now you start seeing, this is important as well, because then let's imagine that you're running, and it's something that I've seen, you are acquiring data from bacteria, and you already know from your previous work, or you did you are checking literature on the work that you're doing, then probably you are familiar with some molecules that are found, so you expect some. If you didn't find them in the library search, but you are sure that they are part of the library is on GMPS, then you know, oh, maybe the conditions that I did were not useful for this, or maybe the extraction was not efficient, or maybe the bacteria, or maybe the samples that I have don't contain these molecules. And that's the way you start finding the molecules. Now, one of the questions is that many might have, and if it's one of your questions, you can ask the same. It's like, how do I know if the molecules that I have in these data sets are microbial? But you can ask the same question as what of them are plant metabolites. As you know, one way it would be like this one. So I also put the link that Simone Sufa, which is one of the leading actors on the MicroMass tool. We did a MicroMass workshop specifically on MicroMass, so you can see the tutorial as well. But we are making more tutorials because there are some updates on the tools, but you can see this really nice talk that Simone did with them and seminars on YouTube as well available. So what you can see here is an overview of how this was built. Is basically you have all the repository that is available in GMPS, but you also have associated information. We knew which files or which data files were obtained from monocultures of microorganisms. We know this file is coming from an Staphylococcus aureus on these conditions, and then we have the blank of that culture media as well. So we filter out all the blank signals, so we have only molecules that are coming from the Staphylococcus aureus. So in that sense, every single spectra we have from doing MicroMass, we guarantee that is microbial, even if we don't know what exactly that molecule is. If the molecule has not been identified, but only the spectra is available. That way, it's possible that in the future, some of you might tell me, Mauricio, I found one of the molecules here, and we already characterized the structure, and it's this new one, and it was present in one of the data sets. So we will have two hits as well, because then you find in your project, but you also are able to annotate a molecule that was already detected that we didn't identify yet. And then using the spectral alignment, you will find that the similar as we do the spectral library match, and we saw, okay, this molecule is this one. Well, in this case, what we are finding is the molecule is actually microbial because it was found in this specific data set. What is? Okay. So let's explore the results then. I hope I'm not skipping one of the slides that is there. But here is just to give you a couple of examples where these N-acylamides have been reported as microbial. So since I mentioned yesterday in the talk that we have these molecules that have been reported, we synthesized some, Elena and William, which are leading one of the other projects on annotating and curating a huge knowledge base of microbial molecules. And that is a different approach, which in this case, we are actually annotating molecules that are known from microbials, from literature, getting the spectra, and then using those to build a knowledge base on this. These molecules have been reported as bacterial, so this is relatively new. It can be not that super complex in terms of chemistry, but it's something interesting because we are finding them. So when you use in the library matches on the job that we just did, we just look for histidine. We have at least 6 candidates of these 5 or 6 candidates. Then you will find them just by looking for the word. Then let's grab one of these. So we have this clicking mirror of this. You have in this number, you see here on the column scan, which this is basically the cluster index, which is the identifier of the fragmentation spectra. It seems that it's an acyl histidine with carbon change of 14. So you find here, and this is when you now, for the first time, you know what a USI is. So the USI is a universal spectrum identifier, as I told you, and you see to the right panel, when on top of the fragmentation spectra, you see all this long path, which is basically the USI. So it's characteristic for this spectrum. So you know here, after the task, like this, so you see the task and you see the number. This is the task of the job that you just did, and this is the folder where it contains, and at the end you have the number. So you see the 64239, which is the scan, which is the spectrum. So now you are comparing this against the library annotation that we have from this molecule. So we know at least that it's an acrylamide. And then we are going to use that USI to use the micromass. So that is how you start using the USI. So you can search every single spectra using that path. That's how you run the molecular network. You got all the USIs for all the spectra. It can be thousands. Then you can grab the ones that are interested to you. So you can search every single one. We have also the batch mode when you want to search several ones at the same time. But for now, just learn that you can do single search. And then once you're familiar with that, then you can start reaching out. We have the code in GitHub as well. And then you can search for several at the same time. When you put the USI on that Spectrum USI link here, then just click by search micromass by USI. There are other options. It's possible that you have papers in literature that have, oh, this molecule was reported in the 90s, and they have the spectrum in the supplementary material. And you can get the spectrum there, get the signals there, the intensities, and then just plot it here at the spectrum peaks, and then just do the same way. But for now, it's just easy way is just put the data into molecular network and then just grab the USIs and then just find it. This is how the results looks like. And this is an hypothesis generating. This molecule has been observed in at least one of the files that is in micromass that is coming from bacteria. And that is a suggestion that is also confirmatory because these molecules have been reported from microbes, and we are also observing these molecules in microbes in reference data sets from this. So if we do library matches in this one, it's just basically showing you more information that we have from this single search. So it tells you where it is, what library matches we already know, but also in which data sets this molecule has been observed. So then you can confirm if that makes sense, if it's rational to have them. Then you can inspect, and you can even reach the collaborator that published those data sets because the contact information is there. And then you can tell them, hey, I found this molecule that is interesting because it's present in my experiments, etc. It looks like it's microbial. And from your data sets, it looks like it's present in yours. This is important, for instance, in terms of structural elucidation at some point, is that it's possible that the collaborators also have high yield of those extracts containing this molecule. If you want to confirm, then you can run some experiments on coelution and then confirm that the molecule that you have is actually real, which in this case we know is real, but that's the path that you should focus on to confirm. And then you have the taxa matches on this. This is all possible because once we curated those data files, we associated all the information that we could. As much information you can associate to this one, the better. And then this is, I just gave it as like a exercise, but I don't know if there is time, because I want the users also, because I think I cannot see you, but you can find more examples. So you can play with the other examples of histidine, or you can find others, but you can find the other examples of histidine and then tell me if you find or you found microbial sources using MicroMast. So I can give you a couple of minutes. I don't know if we have, because it was for 30 minutes, but it's already 43. It's fine. So if you give them, and then let's give it 2 minutes. I hope that anyone that is playing with the tool, with the MicroMast, they let me know if they found more examples. If they struggle, that is also good, because then I was like, oh no, Mauricio, you went too fast and I was not able to find it, or I couldn't do it. But it's basically following the previous one. So it's like, if you go and then just grab from these examples, you see, when I put it like histidine, you see that there are other examples. You can do the same that I did for the 64239. You can use the same approach for the other candidates of histidine, and then let me know what you found, because I also have the answers, but I just want you to do it.
I think that everyone needs the access to Gentest 2, right?
The link that I provided, I think that they have access to, so they can play with.
Oh, okay.
If not, then because then we just follow up. But I think that they can click in the link. They can do basically what I did here.
Okay.
That they can follow up, because that's something that I want. Like, again, like if in a couple of years, some of you that are doing the PhD, or you're working in your projects, and then you find, hey, Mauricio, look, I was using MicroMast, and then we finally confirmed the structure of this one, and it was from these data sets that were already available a few years ago. Then we confirmed the structure, and that will be one of the good news on the usefulness of these resources. So it's one thing. Can you access the link, right?
Yes.
Okay, and then you can see the library matches, and you can go with this thing, and you can go, okay, great. Perfect, everything is working fine. I don't know how to say, vamos a explorar los resultados. How do you say, let's go to explore the results in Italian? I don't know how to say that. And andiamo esplorare i risultati. Andiamo esplorare i risultati. Yama. Instead of ayamo. Yamo. Oh my gosh, you have like so it's like not an accent, it's a different accent. But it's a different word. So completely, right? It's the influence. Yeah. Okay.
Can I ask you?
Ayamo. How is again? Instead of ayamo, llama. Llama. Okay.
True Napolitan with the double L. Yam.
Llama. Llama. Okay. Llama. No, I need to learn Italian then. It's something that we want to, but
Can I ask you something?
of course, questions are welcome.
Okay, so if I understood well, microMAST works also for unknown molecule.
Yeah.
Okay.
The only thing that is you will not have library matches, but you will have if the molecule was present in a data set.
Okay.
So you will have something like this. You will have it can be a bigger tree because in here it's just it was found in Staphylococcus aureus, for instance, which is interesting. But then you might have here in library matches, if it's unknown, then you will not have anything because you will not know an annotations because we don't have.
If it's unknown.
But still, you have where it
Line.
was from.
Okay, but
Exactly, and it will
also,
have.
can I find some microMAST can tell us if this molecule that is unknown is similar to known molecule or not?
This will not tell you, but you can do it because you
Okay.
can do, for instance, the spectral alignment when you have this. So you see, you can grab the other USI
Okay.
from the molecule that you would think you suspect that is similar. But if it's not similar, if it's similar, it might be in the same molecular family. If you run the molecular network together. But if not, you can grab from another one that you say, hey, I think that this is familiar. Grab the USI, put it here in below, and then you will be comparing two fragmentation spectra.
Okay, yeah.
And then you will see if they're actually similar or not.
Or not, yeah.
Exactly.
This I know about.
Yeah, and the reason it can be that the cosine score might be low, but not necessarily they are that different. It's possible that you have in one, let's say you have more noise, and the other has like clean, and then it's like less peaks in the cleanest one versus the other one that is with some noise. And since the spectral alignment here is comparing to spectra, is basically comparing the entire spectrum. So it's possible that the cosine score is not that big, but it might suggest which fragments are the same. And then you have.
And also I have another question. Can be an idea, because we have a lot of data also about gene cluster, to correlate the molecule.
About gene clusters?
Yeah.
Okay.
To correlate the molecule with the gene cluster. So, for instance, I don't know, it's an idea. I think in also
Yeah.
in the future, maybe it could be nice to see if the molecule derives from bacteria, of course, but if which gene cluster is responsible for this molecule.
Yeah, but it's something that is not known. If you think about how many molecules are known.
Yeah, I know.
It can be 300 to 500,000. Let's assume. I think that is in that number. How many of the microbial molecules have been linked to their BGC? And I can tell you that is less than 100.
Yeah, no. It's I mean,
with that
there's
is work
a
to do.
MIC database is a little bit huge. I mean, there's a lot of molecules correlated to gene cluster.
Yeah, the MyBig is one. The anti-smash is based on the MyBig, but then it's like
I know
hundreds.
it's hard. I know
Yeah.
it's hard work, but yeah, maybe in the future.
It's still ongoing.
Yeah, it would be nice to see. Okay, this molecule is unknown,
Mm-hmm.
but this molecule is similar to this molecule that is known,
Yeah.
and I know that the gene cluster of this molecule is this that is from this type of this strain and so on. I mean, I know it's a dream,
that is
but
that is something that we need to Yeah, exactly. That's why it's important to have these microbial molecules. Like the more
Yeah.
molecules are identified, or even if not identified, at least we know that they are microbial. We can focus on, okay, now that we found and we confirm that it's microbial, let's find the biosynthetic gene cluster.
And also it's important
Because
to have
that's-
data.
Yeah. Okay, how
Uh,
are we going? Because then it's 2 minutes. I gave them 5 or 7. Did they finish? Okay, so because I can go on and then just give you some answers, but I hope that they were able to do it. So just to give you an example. Okay, from the task that I say, okay, let's search this one. That's the 75302, which is a candidate of histidine, but with 18 carbons and a potential double bond. So when I run this, it looks like it has at least in one bacteria, but it's not additional information, but it has in fungi. It was like, okay, it's found in some of them. So you can have more information on those. So you can even, again, you can go to library matches, you can go to data sets, you can see more information if it's there available. The one thing that definitely is true is that we have library matches because we are looking for a molecule that has a library match. This is one that you should have seen this. Or example, if we grab this one, which is with a long chain of 12 carbons with no double bond, and it looks like this is also from a fungi. It's like only one. So this is also a hypothesis that you have, but at least we know that this molecule definitely are microbial. And let's find this one, which is with a 16 carbons, and then with this, we found that is coming from Pseudomonas, which is also a bacteria, which is also some of the Pseudomonas have been studies well known, similar to the Bacillus subtilis as well. But you see that we start finding molecules that we didn't expect it that were there, and some of the molecules have been actually identified previously, but it was just the chemical complexity was not that big, and people want something like more complex for novelty, etc. But these molecules are important, and then you see how you start finding these links of molecules. And again, to your question, is really good because then we can focus on finding the biosynthetic gene clusters that are associated to these molecules to continue refining, which is something really missing. And that's why I still like to keep working with microbes, because there are many molecules that we keep finding, and so on. Now, the final thing that I want you to do it later on, now that you are masters of microbe mass, you know how to look for molecules, is that you can explore the whole results. And this is the way that you basically explore the network. And then you can just do the library match here to visualize which molecules are annotated from your entire data set, and then focus on those as a starting point. And with that, we covered the micromass analysis or exploration of results. And I think that is a good starting point for you to just working on your data sets. You know how to mine public data sets, but now you have the tools of how to analyze the data that you are generating. And of course, feel free to reach any of us. I'm happy always to collaborate with any of you. And I have more resources there. Here I put also links to the seminars that we are working with Daniel Petras, Allegra Aron, and Ning Wang, that they have a lot of seminars and tutorials for each of the tools that we are working on in applications. And if you think that there are many others that are needed, and of course, feel free to reach. And I'm directing this MS Collaboratory when we're working with aquatic symbiosis. So if you are interested in collaborating with us, just I provide the link of the website that I'm building as well, and then feel free to reach out to me and we can discuss how we can collaborate as well. More tools, and then you have a lot of the good thing of this is that we have a whole community that's willing to to collaborate all the time. So yeah, feel free to reach out any of us, and thank you. With that, yeah. Oh, thank you. I was not expecting the applause. Thank you very much. If there are any questions, or I don't know if we have more time, or maybe we have for the next workshop because it's too much. There were questions, or people were busy following?
Yeah.
So everything was clear. I hope.
Yeah, I think it was really clear, and if you, of course, it's very nice that you give them the possibility to reach you out, because sometimes you're just thinking, and then when you are back in your lab, you say, "Oh, I have this to ask," and this is very nice to be able to reach you. And also, I would like to say also to other to people that are online that we are going to have the in-presence edition of the Summer School on Natural Products that will be in Maratea, Italy, on June 29 to July the 4th. And Mauricio will be with us, of course, so that you will have the possibility to interact for a long time. It's 5 day schools. We do a lot of interaction activities, and so I think that this will be the chance to give more, and of course will be of great helpful for all students. So please have a look, follow us on our website that we are going to show. Perfetto. Double T. Perfetto.
Grazie mille.
Grazie a te. Okay, so follow us on our socials. So you have the website of the school, and where we will soon post all the program, and you can also register through the website, and you also follow us on Instagram, where we post information. And then sorry, I would like finally to thank all the speakers of this edition, and all the organizing committee, Viviana Di Matteo, Teresa De Rosa, Paolo Scogliamiglio. Special thanks to Germana Esposito, that was the real power of this event, and that managed all the logistics related. That was not really easy because, of course, we are abroad, and also all people part of the roundtable chairs, and all the participants. So hopefully we will see most of you Maratea next year, and I think that's the end. So please fill out our satisfaction questionnaire. You can give us suggestion, advice for next edition, and if you need the participation certificate, please write us by email. So thanks, and see you next edition. Okay, now the lunch is ready.